Who here has heard of rabies or Ebola? Who here has rabies? Good. So these are diseases that start in animal hosts and become infectious to humans via pathogens, which are disease-causing agents. So bats are the known reservoir host of rabies and the likely reservoir host of Ebola. So that means that they have the pathogen inside them, but they they can still infect individuals, but they don't show symptoms. So they can still infect humans and other animals. Studying wildlife diseases is very important because we can effectively mitigate and control these outbreaks before they become pandemics like Ebola. So I'm interested in studying pathogen transmission in little brown bats. So bats, to save energy, can choose high-quality roosting areas, cuddle with their little bat friends, or enter torpor, which is a state of reduced body temperature and reduced metabolic rate. So it's like a super sleep, if you will, which reduces activity levels, which would likely reduce their chances of coming into contact with other individuals who are infected with a pathogen. Habitat selection can also affect the likelihood of acquiring a pathogen. So if there's one high quality roosting area in an area of poor quality habitat, individuals are going to congregate to that area and are more likely to come into contact with an infected individual. So to study these hypotheses, I captured 10 bats outside of a cave in Manitoba. I affixed temperature data loggers to the backs of their bats, which recorded body temperature, so I could notice when they entered torpor and how long they were in torpor. I put all of these bats into an outdoor flight tent, which had 4 roost boxes inside of it, one of which was heated to cause that aggregation. To study pathogen transmission, I infected one of the bats with the deadly, highly contagious pathogen. I'm just kidding, I used UV fluorescent powder. It was lime green. I put one infected individual in the tent with the other 9 individuals and allowed them to fly around for 24 hours and infect or hang out with each other. I recaptured all 10 bats and took photographs of them under black light, so like a mini bat rave, and then released them all back into the wild. So I did this 15 times with 10 bats each for a grand total of 150 bats. For each of the 15 trials, I had data on their roosting group size, how much they used torpor because of the temperature data loggers, and I also had data on how much of the pathogen they had acquired or how sick they were. Preliminary results showed that the warmer the roost, the larger the group size, but the larger the group size had no effect on their likelihood of acquiring the pathogen. So remember, if you're a bat, it doesn't matter where you roost, it matters who you're roosting with.